In this lecture, we will cover traditional culture and identification methods. When identifying the bacterial organisms, you use various algorithms and identification schemes or flowcharts to help identify these organisms. And this is based off things like colony morphology, how it grows on the plate, the microscopic morphology, if it's gram-positive, gram-negative, cocci, rods, chains, pairs, those things. The biochemical characteristics, so these are some of the spot tests or different biochemical tests that are performed. And the most probable identification is based off, there's different variabilities within the organisms and how they metabolize, or the different phenotypic type characteristics. So the organism may be the same genus and species, but there could be some slight variations as to how it reacts with some of these different test methods. There are some that have slower reactions or delayed reactions, such as they may ferment lactose a little bit slower than another organism that is the same genus and species. So maybe cultured from one person, it gives a certain characteristics, whereas it's cultured from another person, it gives a different characteristics. And the probability of positive versus negative reactions, so you kind of have to use some of those characteristics. If it's fermenting slower, then you have to base it off the probability of it actually producing a positive reaction versus a negative reaction. So when setting up a culture, you are streaking the plate to try to get isolation. So you are typically using a 4-quadrant method where you start in the first quadrant, you streak it, and then you turn the plate. You go back into that first quadrant a couple of times and streak it out. And then from there you will do another streak and then the 4th quadrant. With these, what you're looking for is it starts off with a thicker growth pattern, so you can see here at the bottom, and then it gets a little bit thinner, and it thins out till you get to the point where you actually have individual colonies growing. And so if you look at this video here, you can see where the I can get it to play here. So here in this video you can see where you start with that thicker streak, and then it kind of gets streaked back into, and then you can start to get some colonies growing. And as it streaks out, you can kind of see those little lines as that plate tilts. You can identify bacteria based on a genotypic criteria. And so this is when the bacterial genome is characterized using a molecular technique. And so with this, you're looking for the presence of a specific gene, and this is a definitive identification of the organism. When you look at the phenotypic characteristics, you're looking what's observable physically. So you're looking at it microscopically, you're looking at how it stains, what it looks like under the microscope. You're also looking at it macroscopically, so the growth characteristics of it on a agar plate and how it grows with different media. You're also looking at the metabolic characteristics, so this is the growth requirements, the susceptibility testing to different antibiotics, and also some of the nutritional requirements and how it metabolizes various things. So one of the main things that we look at when we're trying to determine what an organism is we're looking at the colony morphology on the agar plate. And so with this, you're looking at things like size, you're looking at the color or the pigment of it, the shape of it. Is it elevated and raised? Does it have irregular borders? Does it have any special characteristics to it? How does it look for the surface of that colony? Does it glisten? Is it transparent, translucent? Is it dry? Is it dull? Is it opaque? Meaning it actually does have a color that you cannot see through. Does it look wet? Does it look mucoid? You're also looking to see if it changes the agar in any way. Does it hemolyze the agar? Does it change appearance due to some of the nutrients or different indicators that are in the agar? Does it pit the agar? Does it do anything different to that agar? And you can also use the odor of the organism growing on the plate to help characterize it. So when we look at colony morphology, we're looking at the colony as an individual, so not as a group. So these up here are mixed and you have several in a group. We're looking at them individually. So we're looking at the color. So you have some that are white. You have some varying sizes, but these are all pretty small. You have some that are transparent. So these are transparent. With this one here, it is 2 colonies that are side by side, but they have also did a partial lysis of the agar, of the blood that's in the agar. And so that would be a alpha hemolytic organism there, and since it has had a partial lysis, and so you can kind of see sort of a green tint halo around it. It's kind of hard to tell if these are raised or flat, but just from what I remember when I looked at this, the white colonies are raised, the hemolytic ones are, or the transparent ones are a little bit flatter. And so with looking at the elevation, depending on the organism, that helps you to determine a little bit about what organism it may be. And you're also looking to see if they have any sort of irregular borders, if they're round, if they're teeny tiny, kind of pinpoint, if they kind of do like a swirly type action. So you can kind of see that here, and we'll have an example of that here in a little bit. So you're just kind of looking at different characteristics of each individual colony. When we look at color, again, you have that hemolysis of the agar here, but these organisms are kind of grayish, transparent, and then you have some that are white. These colonies up here at the top, they all kind of have a whitish gray look to them. You have some over here that are kind of a yellow, or some people may say buttery. And with these, you can also kind of see a little bit of a raised center to them. So it gets a little bit darker in the center where you have more organisms in the center of it, so it kind of has a little peak. And then of course the rest of these kind of have more of a white look to them. You can see these colonies at the bottom pretty good, that they are a little bit raised. And so when you have different organisms, you can kind of, based off how it looks growing on the plate, kind of start narrowing down what the organism is. And you'll learn these characteristics as we get into the next course of microbiology and focus more on the bacteria. With size, you are looking to see if it's a large colony, medium, small, or pinpoint. And so here at the top we have some that are considered small. So you can see just in relation to the size of the colony on the plate. And so you're looking at them again as individual colonies, not as grouped together, how you have them kind of grouped in the center. And so this second plate here, they're a little bit larger. So these would be more of a medium-sized colony. And again, you've got the thicker area, and of course when they're kind of growing side by side, they look small, but once you get them out to themselves, they're a little bit bigger. And then it streaks out, and then you have another streak I can't quite tell if this is just a 3 or 4 quadrant street. Then your next plate here, you have some large colonies. And then if you look at the video to the side, you can barely see those colonies, so you really have to tilt the plate to see them, but those are pinpoint. Next, we look at the shape. So again, you have rounded shapes. So most of these over here that are kind of a little bit of a yellow tone are round. And you can also see some homolysis here with this plate. So you have a beta hemolytic organism here. And then if you look at this other image, you have some smaller colonies that look fairly round. Picture's a little blurry, but they look fairly round. But then these larger colonies are irregular, so you can kind of see a little bit of an irregular shape to those. So here is an example of a culture that has a metallic sheen. So you can see as you move it, those colonies are very rough, but you can see sort of a reflective metal look to it. And some of these particular organisms will actually have a little bit of a color to that metallic sheen, so it'll look very iridescent looking. So here you have an organism that they said was a gram-negative. They couldn't remember what it was. It has a little bit of a different look because you can see a little bit of roughness to it, sort of dry looking. But what's unique about this, which is sometimes hard to see, but you can kind of see here in this bottom image, and this is what I want you to see, is where it's kind of pitting back down inside of itself. And so a lot of them are doing it where you kind of have the raised edges and then it kind of pits back down inside of itself. But the inside of these colonies look a little rough. And so this is kind of a unique look that you don't see very often. Alright, so here we have something a little bit interesting. So out here in this portion we have the organism that we looked at that had that metallic sheen. But here at the bottom, I want you to kind of focus on this, if I can get this to play at the same time. And I want you to notice how it kind of swarms out. So it starts in a pattern and then it kind of swarms out. So you can see how it kind of has little ripples or waves in it. And then that other organism, you really can't see that metallic sheen very well on this particular plate with this particular organism. And then this plate beside it on the McConkey, you can see that swarming as well. And so this morphology would be considered swarming. With these organisms, you have a mucoid look. And so these are the same organism, but where I mentioned earlier where you can have some variations in how the organisms respond. So if you tilt those plates, you can see how mucoid look those organisms appear. And you can see where it's also fermenting that lactose, so you have that real pretty purplish color with all of these organisms. Urine cultures are cultured in a way that allows you to determine the number of colony forming units per culture. And so you do this by using a calibrated loop. For most urines, you would use the 1 microliter calibrated loop, and you would dip that into the urine, and then the urine that is left inside that circle would be what is actually used to do the culture. If it was a suprapubic urine culture, so this would be where a needle and syringe is used to go directly into the bladder, and that would take a little bit of a larger loop, which is a 10 microliter loop. And then once you have done the special technique for performing that culture, then you would count the number of colonies that actually grew out, and then you would report that out. So for example, if that plate grew out greater than 100 colonies, then you would report out greater than 100 times 10 to the third colony forming units. And of course, this is just a regular culture with the 1 microliter loop. Or it'd be 100,000 colony forming units of the E. coli. So there are a couple of different ways that this is performed. It will depend on the laboratory that you work at as to which method they prefer. But you have the one technique is to, once you get that loop full of that urine, you would do a streak down the center, and then you would do a back and forth pattern. Some labs will actually do an additional streaking pattern across it. So it will just depend on where you're at as to what they will actually require. And so you can see here where these organisms have grown out, and you have individual colonies. So you would actually count or do an estimate of these colonies. So if I were looking at this, I would get a picture of about 5, what they look like. So I would say 5, 10, 15, 20, 25, 30, 35. But there's many small ones there. So you're looking at greater than 100. So I wouldn't sit and count every colony. I would just kind of give an estimate. And so this is something that you will get used to over time. Most labs, however, do this technique here where the urine would actually be placed in the center in a little circle. So you would get it on your loop and you would just make a circle. Then you would get a clean loop and you would go into this and you would streak out. Same here, you would streak out and you would just keep going around this plate with a clean edge of the loop every time, or a new loop. And so with this video here, you can see as I tilt the plate where you can see some of those streak marks where you kind of get like a little indentation on that auger without cutting it, and you can see how those colonies grow within those streak marks.
There are several enzymatic tests that we use in the microbiology department in order to kind of test some of the characteristics of these organisms. Now this chart here just kind of lists some of the more common ones. We often refer to these as spot tests because we'll just kind of do them real quick on the bench and get an immediate reaction type of result, so we really don't have to let it incubate very long. So you may hear somebody refer to them as spot testing.
So with our catalase test, what happens here is that a little bit of the organism is placed onto a slide. And so you would take a stick or a culture loop, place that organism onto the slide, and kind of spread it out a little bit. And then from there you would drop hydrogen peroxide on that organism. And if the organism produces bubbles, and so what happens here is the catalase enzyme, if it's present in that organism, will convert that hydrogen peroxide to water and oxygen. And then as a result, you will see that oxygen to start to bubble up. So here at the top, you have a positive reaction for catalase, and at the bottom, you have a negative reaction for catalase. So you do not see any bubbling. And one thing that can interfere with this test is that blood will actually give a positive test. So if you've ever gotten blood on article of clothing or anything and you've added a drop of peroxide to it, or even on a wound, you'll see that there are a lot of bubbles that are created. And so you want to avoid getting any of that blood agar accidentally from that plate, because that blood agar could give you a false result. So with our staph, the first thing we're going to do is, if we suspect a staphylococcus, then we're going to do a coagulase test. So there are two different types. Our first test would be a slide coagulase, in which we would take some of that organism and mix it with the reagent, and what we're looking for is clumping. So if the organism is positive, you will see clumping visually with the eye within about 10 seconds. And so you can see here at the top where there is a positive reaction, and then at the bottom there is a negative reaction, so no clumping. And so a positive would indicate a coagulase positive staph or a staph aureus. If it's negative, then you may be dealing with a coag negative staph. If the coagulase test is performed in a tube, then this would be indicated by a clot, would be positive. So you can see at the top where you actually have like a gel type clot. Sometimes they may be a little bit smaller, so you would have to rotate that tube to see it. And then at the bottom, there is no clot formation, so that will be considered negative. You also have the modified oxidase test. And with this, you're looking for a positive result of a blue to purplish color, as demonstrated at the top, or no color. And don't mistake the actual drop of the reagent that kind of puts a tinge on the disc as any sort of color change, but you're looking for no bluish purple color for a negative reaction. Concentration susceptibility. With this particular test, you would perform an inoculum of the organism onto the culture plate, and so you would make sure that this organism creates a blanket all over this plate with a lot of growth. So when you culture it, you would use typically a broth suspension, maybe a swab, and you would rub that almost like you were coloring or something onto that plate or an area of that plate. Once you've inoculated it, you would place a disc, a paper disc that has the antibiotic impregnated inside of that disc onto the plate. You would incubate this plate overnight or 18-24 hours to allow that organism to grow. And what will happen is that the antibiotic from this disc will leach out into this agar. And as it leaches out into the agar, if the organism is susceptible, then it will not grow up to a certain point where that antibiotic starts to dilute out. And if it does not grow, it's considered inhibited. And so if that growth is inhibited, and when you measure this from one side to the next, if it is greater than 10 millimeters, then this is considered susceptible or sensitive to this particular antibiotic. If, however, it is resistant, then there is no zone of inhibition, and that organism will grow all the way up to that disc. Novobicin is done the same way. You have that disc, that NB, that is impregnated with that organism. If there's no growth or a zone of inhibition within greater than 16 micrometers, so from one side to the next of that growth pattern. And these little marks here are just the culture streaks. That's not actual growth, so you can see the more cloudy edges where that organism is actually growing. And so if you measure from one side to the other with that growth pattern, greater than 16 millimeters is considered susceptible or sensitive. And then if you have growth all the way up to the disc, then that is resistant. If you're checking for methicillin resistance, there are a couple of different test methods that are used. Using the disc similar to the last couple of tests, you can use the oxacillin disc to check for that methicillin resistant. So you can see that disc here up top. Or you can also use the cefoxitin disc here at the bottom. And what you're looking for is an organism that is resistant, so you have growth all the way up to it, and an organism that is sensitive. And so if that organism is resistant to that particular disc, then it is considered to be resistant to methicillin. And so this would be where you would determine if this organism is MRSA, or methicillin resistant Staph aureus. And so in that case, most often this organism can be treated with vancomycin, although there are some Staph aureus species that have started to develop a resistance to vancomycin as well. And so with that, what we would do is observe the hemolysis pattern within our culture on our blood agar plate. And so a lot of times with throat cultures or genital cultures, the technician will do what we call is a blood agar stab. And so when you're setting up this culture, when you do that streaking technique, and several of those streaks, you will take your culture loop and actually stab the agar deep down into that agar several times and in several different spots. And so what happens here is that it allows for the detection of streptolysin O. And streptolysin O is an oxygen labile virulent factor that is produced by Strep pyogenes. And so with that, the streptolysin O works best when it's not in the presence of oxygen. So when you stab that agar plate, it gives it an anaerobic type environment to grow in, and it enhances that hemolysis pattern. And so again, just to go over the different types of hemolysis, you have alpha hemolysis, which is a partial lysin, and you have a greening type hemolysis. Beta hemolysis is complete hemolysis, and you get a complete clearing or a yellowing type color. And you have gamma hemolysis, which is no hemolysis. The PYR test is indicated as a positive test if you see a bright red color, so that organism is added to that disc, so you can see the outline of that organism here. And then a reagent is added to it, and it's allowed to sit for 5 minutes. And you look for that pink-red color for positive, and then no color or an orange type color for negative. So you can kind of see a little bit where that culture might be here. And then that reagent is added. So do not mistake that little bit of tinge from that organism and that reagent to be a positive. So for our hypurate test, we're looking for that deep purple color to be a positive, and then no color or a slightly yellow tinge would be negative. So with our CAMP test, what you do is you take an organism that has a beta lysin in it, and so with this would be our organism would be something like Staph aureus. You streak that organism down the center of the plate, and then our organism that we're testing for the CAMP test, we would streak it perpendicular up against that Staph aureus streak. And then after it has been streaked, you will incubate it. And what you're looking for is an enhancement to that hemolysis. And so what it will do will create a bit of what we call a arrow or a campfire type appearance, where it kind of expands out with that hemolysis. And then a negative, you would not see that enhancement of that hemolysis. So this would be considered positive, and this is negative. With our bowel esculin test, we're trying to determine if the organism can hydrolyze esculin. And so with this, what you will do is you will actually take one of your colonies and you will inoculate the slant of the agar. And so once you inoculate it, you allow this organism time to grow. And then what you're looking for is the blackening of that agar as that organism will hydrolyze that esculin. And if there is no growth and no blackening of that agar, that is considered a negative reaction. Blackening is positive. Bowel solubility is used for your strep pneumoniae. And so what you will do is if you suspect that you have an alpha hemolytic streptococci and you want to determine if the colony itself is lysed by the bile or the bile salts, then you would add a small drop to the agar plate. And where you see here that you've actually got nice little colonies, and then that reagent has been dropped onto that colony plate, you can see the remnant of a few colonies here where those colonies have been lysed by that reagent. And so as that colony disintegrates, it can start to kind of leave an imprint of where that colony was at. And so that would indicate a positive test. And then of course if you were to drop it on the colony and the colony remained, then that's considered negative. And so these two here together side by side, you don't have a negative on the plate. Down here it's just where the reagent has not come in contact with those colonies. Your salt tolerance test, or your 6.5% sodium chloride. If you have an organism that is resistant to a high salt concentration, so an example of this would be your Enterococcus, then what you would do is, if you're suspecting that, you could inoculate a broth media with that colony, and then you allow time for that organism to grow. And what will happen is, as that organism is able to grow in that salt media, then it will start to turn cloudy, and if there's an indicator within that media, then that indicator will change from purple to yellow. So this would be considered a positive test. It's really hard to tell that it's cloudy, but it is cloudy. And then this one is negative, there's no turbidity, and that indicator has not changed. Our optogen or P disc is set up just like our other disc test that we did. You create that bed of that organism, place that disc on it, allow it time to grow, and then you're measuring that growth pattern from one side to the next. Greater than or equal to 14 millimeters is considered susceptible or sensitive, and then if it grows all the way up to it, it is resistant. So one of the other testing we can do for our strep species is to do Landsfield grouping. And so with Landsfield grouping, this will classify these organisms based on the type of carbohydrate that is present in the cell wall. And so you will have several different reagents with these kits. And so here you can see some of those reagents. We have A, B, C, D, and F. And you will use these little cards here to test each of those reagents. So if you were to be testing A and B Landsfield groups, you would take a little bit of that organism, you would put it on to that slide, you will mix a drop of that reagent, so the A reagent, B reagent, you would mix them together, rotate them, and what you're looking for is clumping, which will usually kind of float out around the edges here, and you can see a little bit of clumping here inside of this reagent, whereas your negative would kind of be a smoky type appearance with no clumping. And so with your positive result, so for in this example, if it was positive to our A but negative to our B, then we did the Lancefield typing here for our group A S. pyogenes. So Strep pneumo and viridans streptococci do not belong to any Lancefield group, so this test would not be useful for those. And what you're looking for is, once you kind of look at your hemolytic pattern and you're trying to decide for sure if it is one of these particular organisms, then you would test it against this reagent to help narrow down what organism you're working with. With our motility test, we are looking either in a direct wet mount, where we incubate that organism in a broth type media, and then we look at it for that end-to-end tumbling motility for our listeria after 1 to 2 hours of incubation at room temp. Or we can use an agar. So with that agar, we would take that culture onto a needle, which is just basically a piece of straight metal, and then we stab that down into that agar, and you can see here where that organism has grown all along that stab. And then what we're looking for is, if it has motility, then that organism is able to diffuse throughout that agar. So you can see here at the top where you have growth within that agar outside of that slant. You can see the growth coming out here. So the whole agar kind of appears as a cloudiness, whereas this one is clear around those edges outside of that stab. And then of course here you have that umbrella motility from that listeria. So you can see where it kind of forms that little circular type appearance here up top, and then you have that stab mark that comes down and creates that little umbrella look to that organism. So our esculin hydrolysis is very similar to the bile esculin test. The main difference is the tube that is used within your bile esculin is a selective type medium. So they're essentially the same as far as how the organisms present, which is that blackening of the agar. So you can see that here would be positive, and then here we have negative. The triple sugar iron agar, it's also used for your gram-negative organisms, but it can be used for our gram-positive bacilli when we're looking to see if it is producing that H2S. And so what happens here is the organism is inoculated again with one of those straight metal needles, and you would stab down into the agar, and what you're looking for is that blackening within that agar, in which it has produced that H2S. With our methyl red, we're looking for the red color, which would indicate a positive test, or a yellow color, which is negative. So this is our MR. And then here we have our VP. And so with this one, you're looking for this red color, and then this one, of course, is negative. So with the oxidase test, the organism is put onto the filter paper. So it's allowed to sit for a few seconds, and then you drop the oxidase reagent onto that organism. And what you're looking for is this deep purple color, which would indicate a positive result. And then here at the bottom, you see no color, so this will be a negative result. So our spot indol, there are two different methods. Our spot indol is a rapid method. And so with this, what we're looking for is the presence of this tryptonase. And so if it is present within that organism, when we put usually some sort of type of filter paper or something down, we put the reagent on it, and then we take a little bit of that organism
and then we place that onto that filter paper. And then if it's positive, we get that blue reaction. And then if it's negative, you have no reaction. And again, sometimes like some of our other tests that we've discussed before, you can see a little bit of an outline of that organism, and that's okay, but it didn't turn positive. And so if we were using a media to do our indole tests, broth media, then that reagent would be added to the top of that inoculation. So that organism has been put down into that broth, and then we add the indicator reagent to the top of it, and what we're looking for is that pinkish color or reddish color for that positive test, and no color change for our negative test. For a citrate test, we're looking for the organism's ability to use the sodium citrate. And so what will happen with that, if the organism is able to grow in this media and use that citrate, then you'll get a color change, and what you're looking for is this blue color here. And you can see this organism is still a little greenish color here at the bottom, but you're looking for that blue color change to be a positive reaction versus no change for your negative reaction. The DNA test is looking for an organism's ability to produce the deoxyribonuclease. And so there's actually a video that's loaded in the lab section that shows this test, but the method is a little bit different. But he does demonstrate what you're actually looking for. And so with this particular test, when it's set up on this particular media, what you're looking for is the DNA to actually be hydrolyzed. And so what that will do is actually create a clearing of that media around that organism. And so if there is a clearing around it, that is considered positive. If there's no clearing, that is negative. The urease test is used to determine the organism's ability to produce the urease enzyme, which is used to hydrolyze urea. And so with this one, it does produce a color change once those products are broken down into ammonia and CO2, and then you can get a color indication. And if the agar slant was to turn a magenta or pink color or a light orange color, then that's considered positive, and of course no change is negative. The OMPG test looks for the ability of the organism to produce the beta-galactosidase. With this particular test, it helps us to determine a late lactose fermenter from the non-lactose fermenters. We did mention that there were a couple of the Enterobacteriaceae that were the late lactose fermenters, so this would be a useful test for that. If the organism is positive, it would produce a yellowish, orangish type color, and if it was negative, it would be colorless. So here you would see a positive result versus a negative result. Your nitrate reduction test determines whether an organism can produce nitrites, and so microorganisms that are able to reduce nitrite to nitrogen will not turn any particular color, but it will produce a gas in the nitrate reduction. And so this is due to nitrates being converted to nitrites. And what you're looking for is a colorless gas or a colorless broth media with gas production. So you can see that little gas bubble there. This will be considered positive. And then, of course, this is negative. The string test is used to differentiate the Vibrio species from Aeromonas. So with this test, you would actually emulsify the organism into a sodium dioxalate type of reagent. And then what this will actually do, it will lyse the Vibrio cells, and that lysis will result in the release of DNA, and then that DNA can actually be pulled up kind of like a string. You can actually see it with any sort of like stick or inoculation loop. And so to be positive, it would actually have to form a string, and negative would be no string. If you ever did any sort of like DNA lab in high school, you may have done something like this with strawberries or saliva, where you're trying to do a DNA extraction. And so this would be sort of similar to that particular test method.
So some of the additional tests that are performed would be looking at the different metabolic pathways, and this is whether or not they perform an oxidation process or that they are able to ferment certain sugars. And you will get more detail on this as we get later into the bacteriology unit. You're also looking at the amino acid degradation and how they're able to use some of these products with air or without air. And you're also looking at how they utilize certain types of media and how they grow on those media and the characteristics that they present with.
With your oxidation and fermentation, what you're looking for is to see if these organisms if they're able to either oxidize or ferment different carbohydrate substances. And so depending on which one you're looking for, you would use that particular carbohydrate. So for example, we've mentioned with some of our non-Enterobacteriaceae, their ability to ferment glucose. Of course, most of your Enterobacteriaceae do ferment glucose. So what you're looking for is you would actually take a particular medium that has that particular carbohydrate or sugar inside of that media. And you would take 2 of those tubes and inoculate both tubes with several stabs. So you can see here up top where these organisms have been inoculated with several stabs into that media. And so you can see where you have those little stab marks. And so once that organism is placed into that, then is incubated for up to 7 days. And of course you're looking for any sort of production of acid within that tube. So you would do 2 tubes for each particular sugar that you're trying to test for. So you can see that these 2 tubes here, both of them are labeled A, where you have one tube that has no oil laid on top, and then you have one that does have like a little oil layer on top. And so what that oil layer does, that mineral oil, will actually create an anaerobic environment. And so if there is any sort of color change, like an acid production, then it will produce a yellow color change. And so what our results would indicate is if we're having an acid production that's indicated by that color change, then we know that our organism is able to ferment that particular sugar. However, on the other hand, if it is an oxidizer, it will only be in the presence of oxygen. So here at B, you can see this tube here is an oxidizer because it had oxygen here, and this one had no oxygen. That mineral oil was on top. And so the fact that it needed oxygen in order to oxidize that sugar made it an oxidizer. Whereas in tube A, it didn't matter if oxygen was present or absent, it is still able to ferment that glucose in the presence and without the presence of oxygen. And whereas tubes C, neither one of them had any sort of change with or without the presence of oxygen. So it does not utilize any of the sugar that was in that tube. So these tubes could have all been glucose, or they all could have been sucrose. You would basically test the particular sugar that you're looking for. And a lot of the modern methods today either use analyzers, or they will use a long strip test where the sugars are all actually within little wells within that strip, and you'll load the organism into each individual one. So if, for example, if you're looking for glucose in one, lactose in one, then you're looking for the color change in each of those. So maybe it's yellow in these two, so you know that it will ferment glucose and lactose for both of those tubes. And again, some of these have the same way of looking for oxidation, where oil is actually laid over top of those little wells to see if it will oxidize that sugar as well. The decarboxylase test differentiates the different Enterobacteriaceae from other gram-negative rods. And so what you're looking for is the ability of the organisms to hydrolyze some of the different amino acids. So those amino acids would be the arginine, lysine, and ornithine. And so what you would do is you would put, all of those would be in a different tube, and you would inoculate that organism in it. And what you're looking for is it to hydrolyze those different amino acids, and then it would result in a color change due to a pH indicator within those tubes. So this particular method here would be looking for that purplish reddish color to be a positive result, and then no change would be a negative result. So this last tube here is an uninoculated or a no change tube. So that particular tube, you can see that the color is pretty similar to the negative. So this negative tube, this organism was not able to hydrolyze that particular amino acid. The lysine iron agar is used to differentiate gram-negative bacilli based on their ability of decarboxylation or deamination of the lysine, and their ability to form the hydrogen sulfide, or the H2S. So this agar is made into a slant. It actually contains the lysine within the agar. And so with this slant, you've created an aerobic environment up top, and then the bottom is an anaerobic environment. So when you introduce the organism, you would stab the slant, and then you would also streak the slant. So you would stab down into the agar to the bottom and streak the slant. And so you're looking to see how this organism will act in the presence of the lysine with and without oxygen. If the organism is able to ferment glucose, then it will actually start to create an acidic environment at the bottom, and it will turn yellow. If the organism is able to produce the lysine decarboxylase, then it will actually form cadaverine, and that will actually neutralize the organic acids that happens with the glucose fermentation. And this means that the butt of the medium will actually go back to an alkaline state. So it will actually change that color back to that dark purplish type magenta looking color. I don't have a color to match. So we'll actually change it back. And then from there, if the decarboxylase is not produced, it does not produce that cadaverine, which means it will stay that acid yellow color. But if it does, then you'll get that purple color, or will revert back. If the oxidative deamination of lysine occurs, then a compound is formed that will, in the presence of that ferric ammonium, and a coenzyme, and then what it will do will actually produce a burgundy color to that slant, and so that slant will actually change color as well, and that's because you do have the aerobic environment up top to be able to allow it to do that. If the deamination does not happen, then the slant will actually stay purple. So here into the top corner, here's some images of some of those different things that have happened. So with your very first slant, you have an alkaline environment. So you have the lysine decarboxylation that has happened, but you do not have any sort of fermentation of the glucose. Next, you have a alkaline, and let's see, alkaline and probably an alkaline, but it does have the H2S production, so it has turned that agar black. And then our next slant, we do have an alkaline. You can see that little bit of magenta color up top with an acidic. So there at the bottom, that has turned to an acidic. So it has actually fermented the glucose. And then with a D, this one has actually turned red, and then you have an acidic butt. With that, it has lysine deamination, and it has glucose fermentation as well. And then this last tube is no change, it's not inoculated. So this would be what the tube would look like when you start. Triple sugar iron agar is used to detect if a gram-negative rod can ferment glucose and lactose or sucrose, and also whether or not it can form the H2S. And so this is used to differentiate the different members of the Enterobacteriaceae from other gram-negative rods that can be found within your specimens. And so this particular agar contains lactose, sucrose, and glucose, as well as some indicators. And if glucose is the only sugar that's fermented, then there will be a small amount of acid production that is produced on the slant that will be oxidized to a more neutral product. So it'll go from an acid appearance, which would be yellow, and then it would actually go back to a red appearance, just where it would be in the presence of the oxygen, and it would remain at an alkaline type state. Whereas within the butt, there's no oxidation, so it would not revert back, so that butt would remain yellow. If lactose and sucrose are fermented, then there would be like an overproduction of the acid, so the slant as well as the butt would all turn yellow. So here at the top you have an uninoculated tube in tube D. So you can see how the tube would start, so it'd be pretty obvious if that tube color did change. Then from there, if the specimen was alkaline, or if there was no change in the slant or in the butt of the tube, then you have a non-fermenter of glucose, lactose, and sucrose. And so that particular presentation would be here in C, where you have an alkaline. So you don't see any sort of color change. An alkaline acid, so you have an alkaline slant, acid butt. This would indicate glucose fermentation only. So you would see this in this second tube, tube B, where you have alkaline acid. So you have just the glucose fermentation here, not at the top. You can see a little bit right here on that slant where that is still alkaline, but you have the acid at the bottom. And of course this one has that H2S production because you have that blackening there. And then if we're looking for an acid-acid, so of course this used all three. So you get that acid production in the bottom as well as on the slant. And then you can also see where this particular organism produced gas. So you have a break in that agar. Depending on the organism, sometimes it will actually just kind of shred this agar and it'll be broken up all the way through. But most of the time you'll just have a little bit. Sometimes you can have an agar slant that will, excuse me, it'll actually push up from the bottom. So there will be like a little layer of air at the bottom, and then all of the agar would be through here. The carbohydrate utilization test is used to help identify mostly your Neisseria species. And what this test does is uses the different carbohydrates, glucose or dextrose, as it's sometimes referred to, maltose, lactose, and sucrose. And these sugars will all have, or carbohydrates will all have a phenol red pH indicator in it. So if it is, the organism is able to utilize it, then it will change the pH, and then you would get that yellow color would be demonstrated within that particular tube. So I think in one of the previous lectures we talked about testing these carbohydrates on a tube test. And so here you've got an example of a little bit more of a modified test where you have each of these little whales that have a one of these carbohydrates in it. So you can see here C is control, and so you've got each of these little whales. And then here you have your glucose. It turned yellow, so this would be considered positive. Your maltose is yellow, positive. Lactose is positive, and then your sucrose is negative. You also have a DNA test as well as the beta-lactamase test. But for this particular organism, because it utilizes all the way through the lactose, then it would be the Neisseria lactomycia. So with this, you would also want to avoid putting these into a CO2 environment because that can affect how these organisms utilize these carbohydrates. With your X and V test, there are a couple of different methods. This particular method here requires the use of a nutrient type agar that does not have any sort of X or V in it, so there's no blood in it. And what you do is you actually do one of those lawn sweeps where you culture the organism over the entire plate, and then you place the strips down. And so each of those strips, the little pointed end, has one of the factors in it. So this top one has V, X, and XV. So this particular organism has growth around XV. And so this is considered a positive growth, or it requires X and V in order to grow. Then here in image B, we have V here to the side, X, and then XV here up top. And this organism grows around V and XV, so it requires V to grow but no X. And then here at the bottom, well, you got growth all over this entire plate. So this is considered a negative because it did not require X or V to grow. It grew whether it was there or not. But this is what you're looking for. You're looking for the growth in the presence of X and V, or X and or V. Multi-test systems. So these are manual or semi-automated type of procedures in which we perform an inoculation, incubation, interpretation, and result reporting of a particular organism. There's a shorter incubation and reaction time than if we were to do it in a tubed type media. It uses biochemicals and enzymatic reactions, and it is more reproducible and consistent results by using some of these systems. Now, example of this system would be something like the Biomerex API systems, and so there are a couple of different systems here that are used, but basically you have these little small microtubes of dehydrated substrates, and what you do is actually make a bacterial suspension, and then the test kit, dictates exactly how much of that bacterial suspension is needed. So you have some things that you put a certain amount of bacteria in, and you're looking for a particular cloudiness or turbidity to that solution in order to determine if you have enough of that organism present. But then you'll just kind of place that organism into each of the wells that are located here. So you can see how each of these kind of has its own little well. So each of these is an individual test. These here are various carbohydrates. So there's some of those carbohydrates that we talked about. So once you make that suspension, you'll put it in here, and depending on the test, you might have other steps that are involved in it. But then you incubate it, you may have to add some additional reagents after incubation, and then you interpret these results based on this color type of reaction. So for example, these sugars that are down here at the end, if one of them is yellow, then that's considered to be the fact that that organism can utilize that sugar somehow. And so these test systems come in a variety of test system as far as what they can detect. So this one here in the bottom looks like it's probably for the Enterobacteriaceae. And then there's other kits that can look for your staph, your streps, basically what it's going to do is all of these different tests that we've discussed are going to be placed within the strip, and then you do those tests. And so you're basically doing all of those tests at one time. And then after you interpret the results of those tests, it's going to give you some sort of numerical code. And then you will enter that numerical code into the database, or you'll look through a book that has those codes, and then that code will actually translate out to a particular bacteria or organism. Now, the detection of metabolic activity for various systems like this, because you do have some of the manual systems, semi-automated and automated systems. So what it's looking for is the change in that metabolic activity, and so it's trying to measure it in different ways. One of the ways would be colorimetric, where it's looking for that color change in pH indicator or some sort of substrate that's going to act as a chromogen, and then it's going to detect it via spectrophotometry. Nephelometry, you're looking for the light scatter, you're measuring the turbidity. This is going to be useful for your antibiotic susceptibility testing, where you're looking for that turbidity or the presence of that growth of that organism in that specimen. So if it's enough antibiotic there to kill it, then it shouldn't grow. However, if that organism is susceptible, then you're going to have the turbidity within that solution. Fluorometry is going to use substrates that are bound with some sort of fluorescent type of labeling substrates, and you're looking for that fluorescent presence if it is actually metabolized. So your automated systems are basically going to combine a lot of this testing into an automated platform. And again, it's looking for that those changes through the spectrophotometry, changes in color, nephelometry, that fluorescent labeling. So it's looking at a variety of different things. Now these automated systems, and there are several of them, again, you're going to kind of make that bacterial suspension, you're going to put it in those plates or those wells. So you might have like a single well, kind of like what we just saw with the API strips, or you might have some sort of larger plate that's got all of these little tiny wells within it, and so you've got them going up and down, across, and you put that suspension in there, and each one of those wells has a different test that is in it, and then what's going to happen is those activities are going to take place, And then somehow the machine is going to have a way to be able to scan each of those wells looking for that color type of reaction or that turbidity. And it's really useful when you're doing the antimicrobial susceptibility testing, because a large portion of your plate will include various antibiotics, and you're looking to see which one of those will actually work. So this is really good. It kind of speeds up the process, and it helps us to get results to the physician just a little bit quicker. Now, a large portion of the specimens that you get in micro will be blood culture samples as well. Blood culture samples, as you recall, are typically drawn with 2 sets for 24 hours, and then a lot of times physicians will repeat those every day or two. Just depends on the physician, really, how they're monitoring the patient, things like that, or what's going on with the patients. But there are a few different blood culture type systems. You have manual systems that are for your smaller volume laboratories. Not maybe like your smaller rural type of area hospitals may have a system like this, just because it is a simpler system to use and it's more cost effective. Your automated systems, basically what they're doing is like with your BACTEC, what it's doing is looking for usually either some sort of like radioactive type of change where something is like radioactively labeled and you're looking for CO2, release of CO2 with that radioactive label, or CO2 is being measured via some sort of infrared spectrophotometry, or even with your newer versions of these analyzers, it's looking for a fluorescence type of reaction, looking for the release of that CO2 or any of the oxygen that's been consumed within that bottle. You do have a BACT alert system that's made by Biomerics, and that uses a colorimetric type of technology. And so it's basically measuring the microbial production of the CO2 photometrically. You also have another system that's made by, it's the Versa, that's made by Trek Diagnostics, and it's looking for pressure changes within that bottle to indicate that there is organisms growing.